Further exceptions to Mendelian genetics are going to be those that are what we refer to as sex-linked. Means that the genes for these traits, for these mutations, for these alleles are going to be carried on the X or the Y chromosome, hence sex-linked. Now, XX, female, means that there are two copies of every gene, two alleles. Okay. Now that gene may have multiple alleles out there in the population, but in that one animal, that one female animal, there are only two alleles present. They could be the same, homozygous, or they could be different, heterozygous, but there's only two. For a gene carried on the X chromosome for animals, there's only one. So for us males, whatever is present on that X is all we have to work with. So if it is a recessive trait, a mutation, or something that's just not working correctly for whatever reason, that's all we have to work with. Eye color, you see above my head, in fruit flies. You'll come to hate fruit flies in genetics when you take the genetics course. Turns out eye color should be a nice red, nice dark deep red, nice fire engine red. But it's not one gene. Turns out it's a series of genes that creates a metabolic pathway for the making of a pigment, the transportation of the pigment, the modification of the pigment, and moving it around, stuff like that. So it's a whole series of events that ends with nice, deep, rich red color. Turns out when you interrupt at the different steps of that pathway, you can end with different colors of eyes. Brown, cinnabar, which is a nice cinnamon color brown, sepia, vermilion, white. We know that the genes for eye color are carried on the X chromosome. We have found that most of these mutant eye colors are going to be found in males over females out there in the population. Why? Females have two X's. If one X is carrying a mutation, carrying an incorrectly coded for protein, the other X could still have a working version. Thus, the pathway could continue. Males, we only have one. Even the fruit fly only has one X. So that's all they have to work with. When you go through and you do Punnett squares of the different, sex-linked, whether it's something carried on the X or something carried on the Y. If it's carried on the Y, it's only going to show up in males no matter what. If it's carried on the X, well, it could be a female, that shows the phenotype, the different eye color, the antenna thing, whatever. But more often than not, as it's showing here, the female is just going to be a carrier. The males, since they only have one X, and if it's carried on an X, that's all they have. It's interesting. There are genetic diseases that are male only, or it's for whatever reason, it's carried on the Y, and well, that's all you're going to see is humans that have a Y are the only ones who are going to be able to express this. Just because a male is carrying or a female is carrying doesn't mean, They may not express this disease, this genetic mutation in their phenotype, but 50-50 chance their male offspring will. And it's a 50-50 chance that their female offspring will also be a carrier. So we find that mutations on the Y generally are going to somehow affect progeny. It's going to affect downstream male progeny, and more often than not, mutations in the Y are what they refer to as dominant lethal. It's going to screw up development. Now, it may not be lethal as in killing the offspring, but it can render them impotent. It can interfere with, maturation, development. If it's on the X, well, it's going to be expressed in the male because that's all we have is the one X. For females, 50-50 chance. They're going to be carriers, or they're going to have, be homozygous for whatever that is for it to be affecting their phenotype.